Good morning, everyone. Welcome to today's distinguished Warren seminar. It's my great pleasure to introduce today's speaker, Dr. Audrey Sawyer. Dr. Audrey is currently a professor of earth sciences at the Ohio State University. She's also affiliated with civil, environmental, and geodeck engineering. She got a PhD degree in geological sciences at the University of Texas, Austin. And her study, she studies the interactions between surface water and groundwater, which has important implications for water quality and our ecological systems. She has published over 50 papers, including Science, and she has received many awards, including the Cohort Early Career Award of Geological Society of America. Today, she's going to talk about phosphorus transport between surface water and groundwater. So without further ado, the floor is yours.
Thank you, Judy. It's great to be here. The last time I was here was about 15 years ago as a graduate student working in the St. Anthony Falls lab. And Judy's going to take me out there later today. I'm super excited to see how things have changed. You all have a wonderful campus for doing research in civil and environmental engineering. And it's just a real honor to be here with you today to share my research. I'm going to be talking about a set of projects that we've been working on over the last, I would say, maybe 5 years or so in our backyard, trying to understand the hydrology and nutrient transport processes in heavily modified agricultural headwater streams. And these, I mean, we honestly call them ditches a lot of the time because they have been so heavily altered by human processes. The work I'm about to show you has been the work of a large number of students and collaborators working collaboratively. And it wouldn't have been possible without all the great folks on this slide. And I also want to just give a shout out to the landowners who have been so supportive of doing research on what is sometimes a contentious issue of nutrients and water quality right in their backyards. They've been very fantastic and supportive. As you all know, being in a Great Lakes state, we are struggling a lot of the time with water quality in the Great Lakes, and particularly in Lake Erie. It's sort of a big, broad bathtub, and the nutrients humans have been putting on the landscape, not just in agricultural areas, but also in urban and suburban areas. When it enters Lake Erie and that big, warm, broad bathtub, it creates what looks like pea soup from space because of the harmful algal blooms. And one of the things that has motivated my research and a number of other researchers' investigations in this part of the world has been the trends that we've been seeing in phosphorus over decadal timescales. There have been some changes in best management practices that have helped us start to get a handle on phosphorus traveling to Lake Erie in particulate form, the particle-associated phosphorus, but the dissolved component of phosphorus has been really hard to get a handle on. You can see it was sort of going down for a number of decades and then seems to be going up again. And that brings to question all kinds of unknowns about what is the timing of this dissolved phosphorus storage in the watershed and how it gets to Lake Erie. So for me, as someone who studies surface water, groundwater interactions, I've been really excited by this problem. And what have we been doing? We've been working in these small headwater streams, which are the ones that we often don't get long-term monitoring studies of. Like a lot of what we know about water quality takes place in third, fourth order streams where, say, the US Geological Survey has a long-term monitoring station. In Ohio, we have the Heidelberg network. And what we don't often know about is what's happening up in these little headwater streams. And these are, I would say, arguably one of the places where humans have really impacted the plumbing of the watershed, because you can see all these little red drainage ditches on this database. They're places where there was probably once a sort of natural 0 to first order stream. It's been straightened and it's had the riparian cover, the tree shady cover removed. So it's been turned into something like a straightened drainage ditch. What you also see is that there are these pipes going out into the drainage ditch that are called tile drains, underground plumbing, that change the way subsurface flow paths flow to the streams. And so we wanted to know how this affects nutrients, and in particular phosphorus. A little more on the tile drains. I like to think of them as the human extension of Mother Nature's preferential flow paths. So they're basically man-made macropores that are short-circuiting a lot of subsurface flows straight to streams. In a way, we can almost think of them as the underground extension of streams. And they're, at least in places like Ohio, where the soils are poorly drained, they're everywhere. They're really good for increasing agricultural productivity, because otherwise these soils would be too waterlogged to grow crops. But clearly they're going to have a big impact on the residence times and the transport behavior as water flows towards the stream. And so I'm going to start this talk by looking at a study that we did to try to understand some of that travel time behavior. We wanted to know, what is the timing of phosphorus transport along subsurface flow paths to from farm fields to streams via this macropore, man-made macropore network, and how much phosphorus is removed or retained along flow paths. And so to do this study, we went to a site where the USDA has done a lot of cooperative work with the farmer, and we happen to know the tile drain network. It's rare to have a good map of the tile drain network. If it's known at all, it tends to reside with the farmers. The farmers have those maps. Hopefully this just gives you an impression of how extensive this subsurface drainage system is beneath farms in Ohio. So we worked here because we knew where the tile drains were. What we did was we added a mixture of tracers to basically like a little shallow trench, not even a trench, just like a strip along the field above a tile drain. We added chloride because it is conservative. It's going to travel conservatively without reacting, and it's electrically conductive, which allowed us to take advantage of some cool geophysical imaging to see where the plume would go. We added dissolved phosphorus. That's really what we're trying to learn about in our studies. And we also added fluorescent fine particles that give us a sense of how fine particles move in the environment, the kind of fine particles that particulate phosphorus might be associated with. And we added this mixture above the tile drain, and we looked at how it came out about 30 meters away at the outlet of the tile drain to the stream. And we also used geophysical imaging to watch the plume. We collected some core samples, and we tried to look at where it all went. The core samples, we were basically just sticking tubes in the ground as far as they would go. So we could only retrieve down to the upper, I want to say it was like maybe 15 centimeters, but we figured that would be good for seeing what would happen with the particles. We really didn't know what would happen with them, but we figured they'd get really stuck. So we collected some before and after cores. We did the time-lapse electrical resistivity survey. So this was our geophysical survey to understand where the chloride was going. In these surveys, if you're not familiar with them, you stick out a bunch of electrodes in the ground, and then you have like one electrode that pushes current, one electrode that pulls current, and you use two other electrodes to measure the voltage. And you can use that information with a mathematical inversion, an optimization process, to try to figure out how electrically resistive the ground is. And the idea is that changes in time in that resistivity are due to this electrically conductive tracer getting down into the ground. And by doing the time differencing, you actually can get a much clearer idea of the subsurface than if you were just looking at a once-in-time inversion. So the time changes are really interesting to look at for tracking the solute. What did we see with this test? We were really surprised to see that all of our tracers appeared at the tile drain within like 20 minutes. I mean, this is for subsurface transport, I mean, I know there's a tile drain, but this shows you how much humans are modifying the system. Like this kind of subsurface transport is usually what we would see somewhere like a karst conduit, like a cave conduit system in limestone in Florida. And so this is sort of like the conduit system humans have created in the subsurface and these agricultural lands, allowing really flashy, rapid export of materials over tens of meters. And the fascinating thing is we saw the fine particles. I mean, we honestly never knew if we would see anything come out, but we saw it come out, and we saw it come out within tens of minutes. One other thing that we noticed is that the dissolved phosphorus was not transported conservatively. Roughly, if you look at the ratio between phosphate, the dissolved phosphorus, and the chloride, about 40% of it was either taken up or removed or sorbed by non-conservative processes. We can do a fun little mathematical model to get a little more quantitative information out of these breakthrough curves. This is looking at the breakthrough curves for the solute, the chloride and the dissolved phosphorus on a log-log plot. We can analyze this data using a one-dimensional stochastic mobile-immobile model, where you can think of this as being composed of two different domains: a mobile domain where you have advection and dispersion, diffusion-like dispersion, and some reactions occurring, and an immobile domain where you don't have advection or diffusion, you only have reactions, and things can move across these two domains by some transfer probability. And so you can get stuck for some amount of time, and there's a distribution of wait times within that immobile zone before you get unstuck. And while you're stuck, you can react according to the reaction behavior. And so what we see when we fit our data, first of all, the solutes follow a power law resonance time distribution, meaning that there's this sort of heavy tailing. So you get this initial quick peak, but then this heavy sort of tail of persistence of solute along the flow path that is like persistent. It's ubiquitous in nature. We see these heavy tails in all kinds of systems. In this particular case, we had really strong solute interactions with the immobile zone. So the average time that chloride spent in the mobile zone before getting stuck in, say, the soil matrix was only 16.5 seconds. And the characteristic travel time through the mobile zone, not surprisingly, about 1,200 seconds. You could have seen that from the peak and the breakthrough curve on the previous slide. But what's really interesting is that for dissolved phosphorus transport, the mobile zone was reaction limited, meaning that if you want to sort of remove some phosphorus, you have to get stuck in the immobile zone. You don't have enough opportunity for, say, phosphorus uptake or removal along the mobile zone, because things are just short-circuiting really quickly through the system. And so that creates implications for really flashy styles of transport of dissolved phosphorus in the subsurface in these systems, which is not necessarily something that people usually think about, phosphorus being transported in a flashy way through the subsurface. What happened in the soils, in the cores that we took? This is the before profile, just down to the upper 10 centimeters, and the after profile. You can see we added a lot of chloride to the soil matrix. We added a lot of dissolved phosphorus, especially right near the surface, but note that before we ever did this test, there was an accumulation of dissolved phosphorus near the surface that was probably due to that farmer's nutrient additions to the field. But we did sort of increase that stock. So some of what we added sort of just got stuck behind in the shallow soils. And then the particles, obviously there was none before, and a bunch got stuck after. We can put all this information together in a mass budget for each of our 3 tracers. And so what we see is that if we look first at what's recovered in the tile discharge, so this orange outflow here, about 30% of the chloride made it out in that first day, in that big slug to the tile drain that we added. About 17% of the dissolved phosphorus made it out. That's because, again, of like things like uptake and absorption along flow paths. And then about only 4% of the particles made it out. Most of the particles got stuck in those shallow soils, which is what we would have expected. But it's amazing that we saw any particles make its way through the subsurface and out to these tile drains, which has implications for that sort of cycling of sediments and everything associated with them, every contaminant associated with the sediments you can think of moving from farm fields out to streams through subsurface flow paths. One thing I want to point out is that a lot of the solute was not recovered, and that's because our cores were so shallow. So let's use the geophysics to think about that. These are the inversions from our time-lapse electrical resistivity surveys. So first, if you look at the top, what you're looking at is a map of the background electrical resistivity field before we add the tracer. Our sort of trench that we added it to is that white dashed line. The tile drain is running along the black dashed line. And then 15 minutes into our tracer test, you can see this big sort of plume that lights up where the bulk earth becomes less resistive. And that's because of that chloride moving through the system. Almost an hour in, you can see it starting to disperse and spread out. And then roughly a full day in, you can see it's really dispersed. But what's interesting is that we don't see a lot of bypass beneath the tile drain. So the tile drain is effectively kind of at the lower limit of how deep that chloride is moving in the subsurface. We can look at the breakthrough curves at individual pixels. And so this is kind of a fun little exercise with using the inverted resistivity fields. We can look at, say, location 3 far from the tile drain, 1 and 4 right above the tile drain, and 2 just below the tile drain. And you can see that far away and just below the tile drain, we never really see a change in electrical conductivity. But right above the tile drain, we see a big increase in conductivity, decrease in resistivity during that addition of the solute. And then you see this heavy tail behavior again, which is a lot like what we saw in terms of the like lingering flush of our tracers coming out of the tile drain at the end of the tile drain as well. So here's this persistence, this background sort of persistence of what gets added to the field sticking around in the subsurface. The other thing that we looked at is how much gets flushed in later storms. So we did this addition under dry weather conditions, sort of base flow, tile drain trickling. And then we wanted to say what gets remobilized or flushed during subsequent storm events. And you can see what those look like here. This is the discharge from the tile drain in red and how much rainfall occurred in blue. And the big thing that I want to just point out here, this is what we got in the initial pulse for our 3 tracers. And we got a little bit of chloride and dissolved phosphorus coming out in subsequent events, but you can't really interpret too much from that because you're activating a very different area of the field and flow paths when it rains than when we just had that strip addition of tracers. But what you can think about is the particles. So the particles, like, there are no particles in the field before we added particles. And we saw, it's a very small fraction, but we saw some particles come out in subsequent storm events. So again, this is sort of this like heavy-tailed behavior of a lot comes out at first, but a little bit can get remobilized or flushed at much later times. The last thing that I want to sort of do to connect the dots on the story is give you sort of a year-long perspective, because this test was very time-intensive. We couldn't go back and repeat it a lot, at least over the time scale of what we had funding for and what our students are able to do. So instead, we tried to look at how conditions on the field change at different times of year and just qualitatively infer what we might expect. We did our test right at the start of the growing season. This is when soybeans are on the field growing. And we made some measurements of how the permeability of the soil changes on the field over time. So those are these points. The purple is one location, the red is other locations. They're not always all accessible because of the crops, so we tried to just sort of stitch it together. But you can see that the permeability based on these infiltration tests was really low at the time we did our tracer test. And then it increases at the end of the growing season and the end of the summer when the field starts to get really dry and these soil cracks open up more. And then it closes up again as you go into the winter, as things start to freeze. And so what we're actually seeing is a time when the subsurface connectivity is actually probably relatively low on an annual perspective. And there are times of year at the end of the summer where things are dry, crops have been growing and like creating their root network and all of that good stuff, that we expect even more connectivity and potentially more flashy behavior in terms of that nutrient export from field through subsurface out to the tile drain. So for this study, the big things that I want you to take away, one might be a no-brainer to a lot of folks in the room, but I think there's this growing awareness that our old ideas about how nitrogen is a groundwater problem, phosphorus is a surface water problem, we're starting to break down those stereotypes and misconceptions, and this is one study that does that. We really want to emphasize that phosphorus is mobile in groundwater in these heavily modified streams. We need to think about it as a problem that involves both surface water transport and groundwater transport. We showed that there is a fraction of both the dissolved phosphorus and particles that can act as like carriers for particulate phosphorus. There is a fraction of that moves very effectively through subsurface flow paths in these systems. But there's also a large component that lingers in this experiment for days to weeks. But we know from other long-term studies, months, years, decades. And that makes managing and trying to clean up water quality in these watersheds a really long and involved process that requires patience. And it requires us getting started on that problem today so that it can be better in generations to come. So the implication is that we do have this really flashy behavior in terms of P losses from farm fields through the subsurface, in addition to this accumulation in the long run that creates a challenge for nutrient management. Now, once it's in the stream, what happens from there? We also had a student do a project to really understand the transport pathways within these stream channels, which, you know, humans have modified the subsurface by adding tile drains. We've also modified the morphology and ecology and chemistry of these stream systems themselves by taking away the trees that create structure. That changes the vegetation, aquatic vegetation, and the ecological community in streams. And what I'm going to tell you in this story is that it has all kinds of knock-on biophysical effects on phosphorus transport and solute transport in these systems. So in this part of the talk, I'm going to answer this question of, once phosphorus is in the stream, what is the role of getting stuck in immobile zones, in particular in vegetation, on what is the role of that in the downstream transport of phosphorus? And so the approach is to add the same set of tracers to a stream, look at sampling the water, the stream bed sediments, and the vegetation to understand where those tracers go, and repeat the experiments. In this case, we were able to repeat them over a few different seasons to test for the seasonal differences. In this case, we had an experimental reach with its sub-reaches sort of laid out here at a slightly different farm site. And we have some reaches that we used for modeling purposes and other reaches that we used for just trying to understand the phosphorus transformations on a larger scale. And what we really wanted to understand were these seasonal biophysical controls. And hopefully you get an appreciation for this to some degree by just looking at these three photos. In May, we've got this sort of really thick, freshly growing reed grass that starts to choke the stream. And by December, you can see that things are really dry and withered, and the stream starts to open up again a little bit, especially as we regain that fall flow through the stream. So we wanted to understand the physical effects on the flow by creating zones of low velocity and immobile zones in the stream. We wanted to understand the biological effects in terms of uptake of soluble reactive phosphorus. And when I say uptake, I don't just mean by the plants themselves. I also mean by all of the biofilms that can grow on that submerged vegetative surface area, right? So I, as someone who got my training with porous media, I like to think of these vegetation stands as being a porous media, and all of a sudden, wow, look at all of the new surface area we get for biofilms. So in that sense, you're providing an opportunity for more microbial uptake of soluble reactive phosphorus as well. And of course, the ability to interact with particles and surface area. And then we wanted to understand the physical effects on our particle tracer, like how those fine particles get strained out by the vegetation versus deposited on the bed. And to look at this, we did, again, like a sort of reactive transport model. This one is slightly different. Instead of having the stochastic random walk approach, we had like a traditional USGS code for transient storage. And effectively, the main difference here is that things can exchange across the bed, and once they're across any boundary between a mobile zone and an immobile zone. And once they're in here, they tend to have more of an exponential wait time instead of a power law wait time. And so the mathematical model looks a little bit like this, where in the mobile zone, you have advection, dispersion. We didn't include dilution with groundwater inflows, but you can exchange with the immobile zone, and reactions or transformations. In the case of dissolved phosphorus, we have sorption and like a first order uptake rate. And in the immobile zone, we don't have advection or dispersion or dilution with groundwater. We just have exchange with the mobile zone and another opportunity for things like sorption and first order uptake. And when we talk about the particles, in this case, we treat particle capture on surfaces, so straining in the vegetation, as a first order removal process as well. And there's some earlier literature we used to say that that was suitable and has been done in the past. And so we have our breakthrough curves, we model our data. Another thing that we do with our breakthrough curves is we calculate two things that are like classic calculations for decades for thinking about how nutrients travel downstreams. One is the spiraling length. So you can think about this as the average distance, say, dissolved phosphorus travels before being taken up by a first order reaction or sorbed, or the average distance a fine particle travels before being captured or strained out by the vegetation. And then the uptake velocity you can think of as sort of like the demand. So it actually includes, it sort of accounts for aspects involving flow depth and hydrology, as well as the kinetics. And you can think of it like a mass transfer coefficient for the solute or particles, sort of like a rate of removal expressed as a velocity. So what did we get for our 3 seasons and our 2 forms of phosphorus? The big thing that I want to point out first is that the most rapid soluble reactive phosphorus uptake and particle capture occurred in the spring. And you can look at it either way. You can look at it in terms of uptake velocity, which is high in the spring, or spiraling length, how far you go downstream before you're removed. That's low in the spring. And either way, you can see that the spring is the big time where things don't go as far. It's more retentive. And the particles were immobilized or retained more effectively than soluble reactive phosphorus. So if you look at any season, there's about somewhere between like a small fold, like less than 2-fold to 6-fold change between how different the particles move versus the dissolved phosphorus. And that's important for just thinking about the different time scales of different forms of phosphorus moving downstream. So sometimes the dissolved phosphorus movement is going to be strongly decoupled from the way particles are moving that might have phosphorus attached to them. Other times they're moving a little bit closer in lockstep in terms of their transport timescales and behaviors. And then overall, we saw a similar uptake of soluble reactive phosphorus compared to previous studies of forested and urban environments. So these little agricultural ditches on the whole aren't that much different in terms of their ability to retain soluble reactive phosphorus as a lot of other small streams in the literature. The other thing that we're able to tell from these models is how much the sort of volume of that immobile zone is changing over the course of the season. So what I'm showing here is what proportion of the cross-sectional area that our tracers are flowing through, like what proportion of that is the immobile zone. And what you can see is that the immobile zone takes up more area, and in the spring when the vegetation stands are emerging and thick, than it does in the summer. We were kind of surprised by this, and then we kind of see a little increase again in December. That was surprising. But clearly the spring, when things are first greening up, is the time when we get a lot of that like vegetation creating immobile zones in the stream. A little bit more on the particle fate, because this I think was one of the more novel aspects of the study. We thought that there would be some capture on the in-stream vegetation, but that a lot of it would fall out on the stream bed. And I think that's what a lot of our partners at the USDA also intuited. But in reality, what we see is kind of similar amounts of capture on sediment versus on vegetation. And in fact, in the spring, when that vegetation is first greening up, vegetation is actually capturing a lot more than the sediment. And we know this from samples that we collected, of course, in the stream bed after the tracer test. And also we gave the vegetation a haircut and bagged it and like washed the particles off of the vegetation and counted them. And so we can also get at, okay, so more particles got captured in the vegetation in the spring, but we can also get at something that's called a single stem removal efficiency. So like, I don't know, there's like a lot of things that work with a vegetation stand, right? Like in the spring, you have more blades, but they're also thicker because they're first growing. In the fall, you might still have a lot of blades, but they're thinner and withered. So we can think about the single stem removal efficiency within a stand. And we actually got at this in terms of two different ways, from a model fitting exercise and also from just some back of the envelope calculations with how we gave our samples a haircut and counted the particles on them. And the single stem removal efficiency for particles was greater in the spring than in summer or fall as well. I'm not showing you that, I'm just telling you that. But it all makes sense because when we were visually in the field, we counted up like how big are the vegetation stands, how big are the blades, and the submerged vegetation volume fraction as like a fraction of our total wetted reach was greatest in the spring, a little less in the summer, and a little less in the fall. And all of that plays out for how we capture and store fine particles in the stream over seasonal timescales. So the key takeaways for this stream transport study are that there was really high retention of dissolved phosphorus and particles in the spring when that dense aquatic vegetation was growing. And the particles, not surprisingly, were removed or retained more effectively than the dissolved phosphorus. But how closely coupled those were depends on the season. And much of the particle retention occurred on the aquatic vegetation rather than the stream bed. And this finding is really important because at the USDA, our collaborators often talk about these stream systems as being like linear wetlands. And we've created them to be that way by removing the riparian shading and fundamentally changing the flow structure and the morphology of these streams. And so this is a study that kind of looks at the function of these linear wetlands for retaining nutrients and particles. And we can start to think about, do they function in the way that we hope they do for retaining nutrients as we go forward and develop more of these studies? So the last question that I want to look at from these sites is, I've showed you a bunch of stuff where we inject tracers in the field. now we want to think about like how much phosphorus is just there naturally. Forget tracers. Like, let's just look at what's there naturally, what's being stored, and how easily it's mobilized. And so for that, we did a core collection at 2 different sites and over 2 different seasons, where we collected about 5 cores at 2 different sites, just real shallow things to look at what's in the sediment. And I want to start by saying that we also looked at what is dissolved in the stream water flowing along at the time that we collected these cores and how it relates to stream water quality. I hate making a line with 4 points, but I think it's pretty clear that in the summers when we measured soluble reactive phosphorus, it was high while the dissolved oxygen was low. And this is surface water, folks. So like, this is the way it should be in a flowing stream. And this is the way it is in the summer, like really low oxygen in these streams. And we see, not surprisingly, soluble reactive phosphorus going up. It might be because there's more nutrients being applied to fields and coming off, but there's a lot of other things that can cause that soluble reactive phosphorus to go up, including if you have iron oxides in the sediments that are being reduced under these low oxygen conditions, all the phosphorus that's sorbed to those iron oxides is going to pop off and become mobile. Also, you can look at similar trends with oxidation reduction potential. This is just telling you similar things. But I just want to point out, like, these streams, they're kind of reducing in the summer, even in the surface water, which was nuts and interesting to think about. So what did we do with the cores? We did a sequential phosphorus extraction. This was a student who worked in my group who did far more chemistry than our group has ever done before. This makes me look like I do chemistry. I don't do chemistry, but the student who was working in my group did chemistry. And so she did a pretty heavy-duty, time-intensive extraction of different phosphorus pools. And the idea is that some of these parts of the extraction process are pulling off the phosphorus that comes off really easily. This is the stuff that is going to not be so available for transport. We also sipped pore water from the sediments to look at the concentrations of dissolved phosphorus in the pore water. And what I really want to show you is just that there are seasonal differences. There are differences between site G and site S, but at both sites, both seasons, the pore water has similar or higher concentrations of dissolved phosphorus as the surface water. So pore water is a source of phosphorus to the surface water, and that source, even just by diffusion, concentration gradient. So that source can help explain some of those concentrations that we're seeing in the summer that are higher when the water is reducing. Another thing that we looked at, okay, so this is all the extractions. And the biggest thing I want to point out here is that the G site had a lot more phosphorus than the S site in both seasons. And the mobile fractions that were mobilized with just water, the part that's associated with things like iron oxides, those are a small piece of the total phosphorus stored in the sediments. If we just look at those mobile fractions and we do some back of the envelope calculations, is this an important pool? We find that if the phosphorus in those mobile fractions were existing in the upper, say, 10 centimeters of all of these drainage ditches, and let's just say it were mobilized over a year, right? Like, we don't know what's reasonable, but this is a mobile fraction, so it easily could be flushed over different processes over a year. It would represent as much as a quarter of the total soluble reactive phosphorus load out of the watershed into Lake Erie. And so just that thin slice of sediments and just that fraction of relatively mobile phosphorus is probably playing an important role in stream water quality and nutrient loads within these watersheds. So the big thing I want to convey with the study is first that we have these important seasonal variations in storage in the sediments, and we see more dissolved phosphorus in the surface water when the oxygen is low. So that suggests that there's probably a link with reduction of iron oxides potentially, in addition to complicating factors from fertilizer applications though. The phosphorus content in the sediment varies a lot between sites. I didn't show you this, but it corresponds with organic matter content in the sediment too, and the total iron in the sediments, not surprisingly. And if we mobilize just the more easily mobilized fraction, it can account for a lot of the just slow leaching of phosphorus from watersheds over the year. The last thing that I want to do is just mention that our group is always taking on new members, and the most recent project that we are hoping to look at is understanding disruptions to drinking water due to hurricanes Helene and Milton. And if you're interested in this opportunity, please reach out to me. This is building on work that we've done understanding resilience in our public water supplies in response to Winter Storm Uri that hit Texas back in 2021. We're hoping to do more of this sort of climate resilience analysis. So if you're interested, please reach out. And at this point, I'd be glad to take any questions.
We have time for several questions. Page.
Thanks. Great talk, Audrey. So I've got two questions. One is, I'm an environmental engineer, so I always think about treating and getting rid of this stuff. And so with your second part of your talk, it made me think, well, maybe we should be planting up some of these ditches so you can capture more, but then that's going to decrease the DO even further.
Yes.
And so maybe we should be clearing the vegetation so that you can hope to keep them more aerobic.
Yeah.
And so I just wondered if you could comment on that, or maybe you don't even think about those things, but they seem contradictory.
No, thank you for this question. I think about this a lot. And I think that our USDA partners were thinking about these streams as linear wetlands, thinking like, oh, aquatic vegetation is great. We're capturing things, we're removing nutrients, like it's what wetlands do. It should work well in streams. But having done the work we've been doing down in the stream to really understand transport processes, I'm starting to think we don't want these to look like linear wetlands for the reasons you mentioned. We don't want oxygen low. We want sort of good, just sort of interactions and flow. And so to me, the best thing could be to have a little more riparian shading to create more of those natural headwater stream conditions that probably once existed. But I think the verdict is out on that. And we've done some sort of preliminary sampling of just water quality along these streams. And we've found that if there's just even one big tree that a farmer left, it creates a very sort of riffly like structure. Your oxygen concentrations go up again, your pH changes. And so I'd like to do a bigger survey to get at this question of like, what is better from a stream water quality standpoint, having the occasional tree and more of a riffly type of behavior, or having a linear wetland? And I think we need another study to get at that.
Okay, thank you. And then my other question, I'm just curious of your thoughts. Your first part of your talk made me think that the lawsuit that Des Moines Water brought against agriculture in Iowa, it seems like you've got a pretty good case for these non-point sources maybe being point sources. Yeah. And so I just wondered if you had any comments on how maybe USDA is thinking about this from a regulatory perspective.
That's a great question. I don't know that the USDA gets to think about it from the regulatory perspective like the EPA does, but I can tell you my personal thinking is that these tile drains are so frequent that they are point sources. Like you can put a finger on them, but it's almost like the problem we have with leaky underground storage tanks. Like you can put a finger on it, but there's so many of them that at the end of the day, it's still point source like in nature. I think it's very analogous to that, where it absolutely has a source. You can address it, you can tackle it, and you can put a finger on it, but there's so many that it's still ultimately probably better regulated as a non-point source. I don't know. There's probably aspects of both. Yeah, it would be a good question for me to chat with our local EPA about, because I bet they have some thoughts on it, and I don't know.
We have time for a few more questions. Feel free.
Thank you. I'm kind of curious more about this particulate phosphorus. I don't actually know that much. It seems like you're mostly modeling this particulate phosphorus as being absorbed to inorganic materials. Is some of that particulate phosphorus also caught up in organic matter? So these plants, once they die, is that also particulate
yeah,
phosphorus?
absolutely.
Okay.
Yeah, well, I mean, once the plants die, well, maybe I should address a few things here. First of all, the tracers are just fine particles. Some folks have used them to think about the way organic particulates travel, because their density, I mean, they're ultimately organic, they're synthetic, right? And their kind of hydrophobicity and their density and everything in some ways might actually be a better proxy for organic fine particles than for mineral fine particles. But we don't go that far to say too much about what they're the best analog for. We just think of them as fine particles. And that's certainly therefore relevant to how particulate phosphorus is traveling. But I mean, it would be cool to do comparisons across different fine particle tracers. Like, I don't think we want to use these anymore, because when we got started on the study, like our awareness of the issues with microplastics were coming well into view, but it was sort of like, well, these are still good tracers that have been used a lot in the literature. We're going to use them. And now we're sort of like, okay, let's not add any more microplastics to the environment. Although when you look at the loads that we added, it's like nothing. Have you guys ever cleaned the filter on your washing machine and seen how many fibers come out? Like the load we added to the environment is very small, but I think for practical and environmental reasons, we're going to get away from these tracers, and there's other things we can use, like yeast, brewer's yeast, that can also be used to think about different types of fine particles moving in the environment. And that's one aspect I wanted to touch on with your question. But back to the question about what happens when aquatic vegetation decomposes. Yeah, like this is basically a store of phosphorus. And so when it is growing, it takes up soluble reactive phosphorus, maybe from the water and the soils and everything. And then it's in the plant matter, and then it dies. And then when it dies, it starts to decompose. And so some of that does become it becomes phosphorus in organic particles, but some of it will also ultimately dissolved. And a lot of times when we see high soluble reactive phosphorus in our pore waters, we also see high ammonium, and it's all coming from this sort of dissolving of organic matter. So yeah, as things are decomposing and breaking down. So it's ultimately like, it's a temporary stock. Yeah. It's not a permanent way to sequester things.
And if I could add one more to that, because you showed this plot in the very beginning of particulate phosphorus declining over the decades, slightly. Yeah, do you see that being mostly this inorganic, you're creating these more anoxic streams, or is it a lack of this organic particulate phosphorus?
I mean, there's been so many papers on these long-term studies, none of them mine. So I can only tell you my understanding of the work that's not mine. But a lot of this comes from our improvements in best management practices and how we keep soil and nutrients on fields. And I think what's frustrating to a lot of people who are thinking about best management practices for farmers, like including collaborators at the USDA, is how we're seeing improvements here. sorry, I should have been pointing at this slide for talking about particulate phosphorus. We've seen improvements here on average, while we have, if anything, started to see some rising trends with our dissolved loads to Lake Erie. And so just keeping sediments on the field is not enough to keep dissolved phosphorus on the field. And a lot of this is what we call the legacy problem with it's out in the environment, it's there. In a lot of cases, I think we've already like loaded up a lot of our sites for sorption that phosphorus can sorb to, and like the soil is at its capacity in a lot of places. And we're just continuing to see now that phosphorus starting to move its way through watersheds and get out to Lake Erie. And it's a long-term legacy of things that we've been doing for decades in our farm fields. And in our towns. Like, I don't know about you guys, but every time I walk around academic quads and I look at how beautiful and manicured the lawns are, at least at Ohio State, I'm like, nutrients, and agricultural chemicals, and it's not all farmers.
All right, we have time for one more question. Are they only online? Okay.
Yeah, I'm seeing the trend lines in both of those graphs, but can you just take a second to explain the massive fluctuation from year to year?
Yeah, I mean, I don't have the water fluxes on there, but this is just metric tons leaving the watershed. So say a wet year versus a dry year will have a big impact. And actually, I need to catch up on this literature, but with COVID and also some climatic fluctuations that were happening in a lot of these watersheds, there was a big change in how the growing season unfolded. I think it was in like 2021, and a lot of fields ended up not being farmed that year. And it's sort of like a natural experiment with what happened in our nutrient loads that year. So it's also like it's just, you know, what happens with climate and farming outcomes and the way fertilizers are being applied in sort of a collective scale, including in our suburban areas. So many things.
Any online questions? No. Okay. Let's thank our speaker again. If you have more questions, feel free to say.